Welcome, everyone. Welcome to Science on Tap, a new season. Thanks for coming. My name is Carol Warden. I work at UW-Trout Lake Station. Science on Tap is an example of the Wisconsin idea, an idea conceived in 1905 that the boundaries of the university are the boundaries of the state. Our partners in Science on Tap include our newest partner, Rocky Reef Brewing. Thank you very much for having us. The Lakeland chapter of the Wisconsin Alumni Association, UW-Kemp Natural Resources Station, UW-Trout Lake Station, Monaco Public Library, and WXPR Radio. Science on Tap is supported through a grant from the Brittingham Fund to the University of Wisconsin-Madison. You have 3 ways to watch. You can join us right here in person as we are at Rocky Reef. You can also watch on YouTube live. And when you watch on YouTube, you can ask questions in the comments box as you go. And there's a link to our YouTube page on our website, scienceontapmonaco.org, as well. You can also watch any event later by going to that same website, and we have them archived there for you anytime. Our next event will be on Thursday, November 7th, and you can go to our website to learn more about that. But we will hear from Dr. Mary Stevenson of Marshfield Clinic, and she will be talking about sleep and health. Tonight we have Dr. Jessica Hua. Dr. Hua is an associate professor in the Department of Forest and Wildlife Ecology. She received her bachelor's degree in biology and kinesiology at Southwestern University, and her PhD at the University of Pittsburgh in ecology and evolution. Dr. Hua did her postdoc at Purdue University in disease ecology. She was an assistant and associate professor at SUNY Binghamton, where she served as the director for the Center for Watershed Studies. Jessica moved to UW-Madison in 2022. Research in the Hua Lab at the University of Wisconsin-Madison utilizes an interdisciplinary approach to addressing 2 main goals. Number 1, what are the ecological and evolutionary effects of pollutants on amphibians in fresh water, such as light pollution, microplastics, road salt, or pesticides? And number 2, to evaluate the role of outreach and citizen science in shaping community perception and understanding science. Something else that Dr. Hua is very passionate about is improving representation in the field of herpetology. Toward this, she and a group have been working on a global women in herpetology book. A chapter in this book is a narrative of describing Jessica's path to science. So check that out as well. And per tradition here on Science on Tap, we have a trivia story about Dr. Hua. So, for the research she has to do, it's important that they collect amphibian eggs as close to when they are laid as possible. This means sometimes being out in the field at very odd hours. Early in her graduate school days, she was doing a project, and one of her populations started breeding at 3 a.m. This would have been fine, but the night before, her now husband convinced her to watch the Blair Witch Project. Jessica was terrified of scary movies, but they were early on in their dating, and he loves scary movies, so she made an exception to impress him. That was a very bad idea. The next day, she found herself in pitch darkness in the middle of ponds, in the woods, and a site also close to a state prison, and she was terrified out of her mind that the Blair Witch was going to appear, even though the scientific reasoning in her brain said this wouldn't be possible. That was completely overruled. So there she was in the woods, terrified that the Blair Witch was going to show up. So what did Jessica do to calm her terrified nerves? A, she scrapped the whole sampling event, went home, and turned on every light in her house. B, she stacked up empty collection containers all around her site to act as an alarm for anything that might be coming up on her. Or C, she called up her now husband George and made him talk to her on speakerphone for hours until the frogs finished breeding. Any guesses? C. Way to go, George. And
Oh, Oh, that I maintain to this day. He deserved that 3 a.m. phone. I was terrified. You know when you're so terrified, everything turns white, and you're just like, I was walking through this pond, and every step I was convinced these are bones. Yep, there's a ghost. Terrifying. Anyway, thank you so much for the introduction, and thank you all for being here. I am very excited to have this opportunity to share a little bit more about what we care about in my lab at UW-Madison. So when I was thinking about how to present our work, I thought I'd sort of do it in 3 parts. I'll start broadly, giving you a little bit about the themes we think about, and then I'll narrow into the specific work that we do, and then I'll end a little more broadly again, summarizing the various aspects of our work. So to begin broadly, right now there are two arms of research in my lab. The first is probably what you might think of as traditional science, so the natural sciences. The other is more social science. So on the traditional, more natural sciences end, we ask 2 questions. The first, how do pollutants influence aquatic ecosystems? And the second, why is it that some communities are more susceptible to diseases than others? And we ask these questions in freshwater ecosystems. On the other end, on the more social science end, we're really interested in understanding how non-traditional resources like art, graphic novels, children's books, card games, how we can use these non-traditional resources to communicate our research to broader audiences. We're interested in questions like, can art improve perception of science? Or can graphic novels increase participation in citizen science programs? So altogether, these 2 arms, the natural science and the social science, I'll tell you a little bit more in specifics as I narrow down. So to do that, I was not allowed a PowerPoint presentation. So instead, I brought you some of the art that incredible artists in my lab have generated. And these pieces of art will help keep me on track to tell you about the science that we do. So first up, hopefully everyone can see this. This is a piece of art that I think represents many of the topics that we study in our lab well, and that would be pollutants and amphibians. So in my lab, we study several pollutants, chemical pollutants, including, thank you, pesticides, road salts. We study things like antibiotics, but we also study sensory pollutants like light pollution. We study their effects on amphibians, and there are many, many reasons that we study amphibians. First, they're really awesome organisms. And second, amphibians are declining worldwide. And so it's important to us to understand how various factors, including pollutants, can influence amphibian populations. So to understand the effects of pollutants on amphibians, the way we sort of do this is we use a tiered approach. The world is extremely complex, so how can we pinpoint the effect of a pollutant on an amphibian? We strip away all of the complexity, start simple, and then slowly start adding back in that complexity. And so to sort of tell you about this approach, I've chosen some of our work on just one pollutant, and that pollutant is road salt. So road salt can be an enormous issue for freshwater ecosystems. They serve a very important role in our safety and traveling, especially during the winter months. I'm from Texas, so actually never experienced driving in the snow until I moved up north, but appreciate greatly the safety that it provides. But when road salts go into freshwater ecosystems, it can cause many harmful effects as freshwater organisms aren't adapted to dealing with the salt. So how do we begin to understand the effects of salt on freshwater organisms like amphibians? And I wanted to show you this piece of art. The first way that we tend to do this is to isolate the effect in very simplified systems. So we will conduct laboratory assays. They're highly standardized, all you have is an organism in a glass dish with a pollutant. What does this pollutant do to this organism? So we began studying this on 5 common amphibians in our area, and we could ask 2 questions. The first, do all species of amphibians respond the same way to road salts? So are they all similarly tolerant? And second, we really wanted to know, are these amphibians threatened by the salt that we might find that runs off into their environment? So at environmentally relevant concentrations, is it lethal to amphibians? So we ran this study, and we found that across the 5 species, their tolerances varied widely. So it was almost 45% difference from the most tolerant to the least tolerant of the amphibian species. So to ask the question, are amphibians threatened? It depends on the species. However, when we looked at the concentrations in the field, we went out, we collected water samples, and looked at what the concentrations of salt were in the field. What we found was that the concentrations in the field were not lethal to the amphibians. So it'd be easy to stop there and say, oh, it doesn't matter, we can put salt onto the road, it runs off, it's fine. However, what happens in a dish is very different than what happens in nature when there's increased complexity. So we began to integrate in that ecological complexity. Salts, they are not the only stressor that amphibians face. And they may be a human-induced stressor, but amphibians face other stressors, and this is what this painting is meant to depict. One other stressor that most amphibians face are pathogens in their system. And so we were curious and just wanted to ask a very simple question. At these environmentally relevant concentrations of salt, yeah, they may not cause direct mortality, but could they still have negative effects by influencing an amphibian's susceptibility to their pathogens? So what my PhD student Nick did was he raised amphibians in sodium chloride concentrations that were really low and environmentally relevant. These were concentrations that you can detect in the field. The amphibians did fine, as expected. But then, when exposed to their parasites, we found that at all concentrations of these really low levels of salt, they became more susceptible to the parasite. So taking a step back, the main point here is that in toxicology, in our field, there's a lot of emphasis on what are the lethal effects of a pollutant. But I think what studies like these show is the importance of considering these lower environmentally relevant concentrations, because while they may not cause direct mortality, they can still have harmful effects by influencing the amphibians' responses to other stressors in their environment, like their diseases. So, similar to before, I just told you about a study where we added in more complexity. But I only told you about a study where we included a single species. In ponds, it's never a single species living on their own. It's often something that looks like this. So in this picture, what you're seeing are multiple amphibian species that are interacting with each other. They're competing for important resources. And in fact, this idea of biodiversity and disease is something that has been hotly debated in my field of disease ecology. And in the next minute or so, I'm going to tell you first how biodiversity relates to disease and why people got really nasty about this relationship between biodiversity and disease. So the idea is that in communities, people started discovering some communities that are more susceptible to diseases than others. They looked almost identical, but you'll go into a pond and there's a complete die-off due to a disease in this pond, but not this pond. And so many people have shown that as a community increases in biodiversity, you get a decrease in disease. And people are really excited about this idea because it provides a clear benefit to why we might protect biodiversity. You protect biodiversity, you get that benefit of decreased disease. Unfortunately, sometimes that happens. That's called the dilution effect. Sometimes you get no relationship, and sometimes you get the opposite relationship, where you have increased biodiversity leading to increased disease. And so to sort of give you an idea of how this happens, we can look at everyone in this room. So let's imagine that, well, we are all the same species. There is a virus in this room. Everybody is susceptible. Everybody gets the disease. Now, in a different scenario, let's increase the biodiversity of this room. Let's imagine that everybody wearing blue, you are tolerant to the virus. So there's a virus in the room. Everybody that is tolerant, the virus goes to you, but it doesn't replicate. So it is a dead end for that virus. So everyone wearing blue is a decoy species. So you draw the parasite to you, protecting everybody else in this room by decreasing transmission. So that's how biodiversity can protect a community from disease. But in the same vein, if you have a more biodiverse community, you might also be more likely to have a community member that is an amplifier or a super spreader. So now, everybody wearing blue is a super spreader, meaning you get the virus, you don't get sick, so you keep going out and you spread your virus everywhere you go. And that way, increasing biodiversity can also increase disease. So we see that biodiversity can have all sorts of effects. And so my lab, we're interested in asking, well, what about pollutants? Can pollutants influence whether we see when biodiversity protects a community or when biodiversity harms a community? And so what we did was, it was a ton of work for my PhD students. They generated these bucket communities that varied in the number of amphibian species. Some had 1 species, some had 2 species, some had 3 species. And what they found, in half of those communities, they raised the communities up in salt, the other half, no salt. And what they found was that whether or not biodiversity protected or had no effect on disease depended on whether salt was present or absent. And so the takeaway from all of this is that people are excited about protecting biodiversity because there could be a benefit. But we argue that if we don't consider that extra added complexity of whether the pollutant is in the water or not, then we can't really know when to protect biodiversity. And so we're advocating for integrating in this pollutant perspective into our ecological studies. So we found that those results, and we got really, really excited. But as I mentioned, in this study, we were working in these little Lowe's buckets, Home Depot buckets, in the lab. It's much more complex than the beakers in the Petri dish, but in nature, there are many more things to consider, like predators, changing temperatures. And so this past summer, this is my first field season in Wisconsin as a lab, we decided to go out to 36 ponds around the Madison area. And what we wanted to know was whether or not biodiversity in these ponds protected communities from disease. So this is a painting depicting more glamorously what we did. It was really hot this year. We worked very hard for a week. And what we did was we collected data on how many amphibian species were in each pond, and then we collected data on their disease prevalence. So if you recall, in the bucket experiment, we found that biodiversity protected communities from disease. In nature, biodiversity actually made disease more prevalent. So it was the opposite effect. So something about the complexity of nature affected the direction of this relationship between biodiversity and disease. And so we started thinking about what that could possibly be. And you may have experienced the situation with the drought up here. But this summer in Madison, we were in an extreme drought situation. And so we are hypothesizing that these extra stressful conditions might be contributing to the effects of biodiversity on disease. That being said, we are one lab. We sampled 36 ponds, and it was exhausting. There are hundreds of thousands of ponds throughout this entire state. to better answer the question of does biodiversity protect or harm communities, we have to expand both spatially across the state and also temporally. We sampled for a week. How does it change across the summer? And because we can't do that ourselves, we have been so lucky to have opened up a citizen science, community scientist arm of our research lab. And so this is where I transition from the hard sciences to sort of the social science aspect of our work. When it comes to community science, there are two questions that we care about. First, how do you get people to care about amphibians? And maybe many of you care about amphibians, but for me, I'll tell you, I came from a refugee family. Outside was dirty. I was terrified of frogs. Why in the world would I go out and care about amphibians? And so that's my background. And so one of the things that we do in my lab is to use art, graphic novels, to educate about the amazing amphibians and why they are so important to both our ecosystem and also the services that they provide to human society. And so we conducted a study where we generated a wide range of original art, and we invited a number of people to visit this art show. We took surveys before and after the art show to see whether or not this enormous amount of effort to put together this art show changed people's perception about art, and could it actually communicate something about science. What did we find? We found, thank goodness, that the art improved public perception of science and amphibians and research. And to our surprise, and in hindsight, not to our surprise, the art show did absolutely nothing for science understanding. In fact, the art show, students who went through the art show did significantly poorer than students that actually read the abstract. And so, taking a step back, what does this mean? It means that these non-traditional resources are great for getting people interested. But if we want to talk about and communicate and teach about science, we have to find another way. And so where did we go from here? We transitioned to a graphic novel. And so I actually forgot to bring the graphic novel, but I had 3 to give away, but I'll give them to Gretchen if anybody is interested. These graphic novels are meant to train 3rd graders about biodiversity and disease, and we studied it in the 3rd graders. Did it get them excited about wanting to participate in science? And did it get them excited about learning and understanding about science? As expected, it got them really excited about science, but it still did not communicate. When we quizzed them, they did very poorly. I think the highest score was 57 out of 100. So we came to the point where we understood that art was great at increasing and getting people interested. But how do we ensure that information is being transmitted, and how do we ensure that the data that our community scientists are collecting are of high quality? So what we are doing now, we have designed a program called Friends of Amphibians. What it does, it's a 3 sort of phase program. The first is using an art show to get people excited about the work that we do. The second are accessible resources. So we develop protocols that train anyone from ages 3rd grade all the way up to listen to frog calls. These resources are available online. We're in the process of translating them to Spanish and other languages. And then in addition to that, we are bringing people in and training them on how to listen to and identify frog calls, and then also to be our first step, our first eyes to detecting disease die-offs. We are currently working with 10 local green space areas in Madison and hoping to expand across the state. If anybody is interested in becoming a participant or contributing to our disease ecology, biodiversity research, I'd love to talk to you afterwards. So, putting it all together, what do we do? From the hard science end, we really want to understand what pollutants do to aquatic ecosystems. We want to understand what causes variation in disease susceptibility. Why are some communities more susceptible to diseases than others? We think about things like biodiversity and pollutants. To broaden our work, we care about using non-traditional resources to communicate and also to encourage people to do science. I wouldn't be here if somebody didn't take a chance on me to teach me about the process of doing science. That's something we care deeply about in my lab group. And with that, I am so grateful to have you all here listening, and would be happy to take any questions that you may have about the work that we do.
Am I on? Yes. We will have Amber walking around with a mic, and if you would, you could just raise your hand. She'll get to you. But if you would wait for the mic, that would be great for our online audience and everybody in the room. Thanks.
So just a question about the salt. So does it, the type of salt, does that really have an effect on what it does to the creatures?
Oh.
That's a great question. Can you guys hear me? No. Okay, here we go. So I think that's an excellent question. Does the type of salt matter? And the answer is absolutely yes. So we have tested sodium chloride, magnesium chloride, and sodium chloride tends to be in the middle. Calcium chloride tends to be the most toxic for the amphibians that we test. And we focus on amphibians, but we also have a wide range of other aquatic organisms that vary in their tolerance to these three different types of salt. So yes, it absolutely matters.
Can plants take up contaminants like PFAS?
That is a great question. It's something that we are working on now. We haven't worked on plants and PFAS, so I'll tell you about two studies. We have worked with fungi and PFAS. We have looked at how terrestrial fungi, so we're looking at oyster mushrooms and also red wine cap mushrooms, and in both cases, the mycelium of these mushrooms can reduce PFAS concentrations, and it happens very quickly within 48 hours. And we're really excited about this, but what we don't know is what is happening to the PFAS. Is it being broken down, or is it just being sequestered? And this actually leads to our second study. In the second study, we were curious about aquatic plants and the ability for aquatic plants to take up heavy metals. And in this study, we focused on duckweed. Duckweed took up copper, and it reduced the toxicity of the water to snails, which are highly susceptible to copper. However, the second part of this study was to ask, what happens during the winter when the duckweed dies? What we did was we allowed the experimental units to dry up, and then we replicated the winter in our freezer by placing the duckweed into the freezer, taking it out, putting it back in and out, and then we flooded the containers again with water. Then we took that flooded water and exposed them to snails, and it turns out that the copper enters right back into the water and becomes toxic again. And so going back to that PFAS study, we know that the concentration of the PFAS is reduced. What we don't know is what happens to it. So will it get re-released back into this system once the mycelium break down?
When you did your original experiments on the effects of salt, did you also take into consideration the effects of salt on certain parts of the environment of these creatures as well? For instance, the things that they are living off from, whether it's vegetable or other animals, where they might be predators?
That's a great question. What we did was we conducted so the answer is we did not do it, but my close colleague did. And what they did was they created artificial pond communities using cattle watering tanks. And in these cattle watering tanks, they replicated a simplified pond environment. It had phytoplankton, so algae in the water column. It had periphyton algae on attached algae. They had zooplankton, and then the amphibians. So it's more complex than what we found, what we tested in the jars. What happened was the salt comes in. The zooplankton are more susceptible to the salt than the amphibian. So zooplankton died off. Zooplankton consume the floating algae in the water column, the phytoplankton. So without the zooplankton grazing on the phytoplankton bloomed. When it bloomed, it shaded out all of the resources, the periphyton, the attached algae. That's the algae that tadpoles can consume. And so what they found was that it caused a trophic cascade, sort of a domino effect, by taking out the most susceptible community member of that very artificial community. So we didn't do it, but there are several groups that have. I believe this group, Bill and Rick Relier, they have also looked at fish communities. And fish completely changed the system.
My question regards, the elevated temperatures that we've been experiencing, especially this year, and how that is affecting the amphibians and causing stress, a big stressor in their environment. And also, as the ponds evaporate, that would concentrate the dissolved salt too.
Yeah. I am thinking about 2 lines to answer that question. So the first sort of, we have not elevated temperatures. With shifting climates, another variable that can be changed is variability in winter temperatures. So one example is something called false springs. We work on wood frogs. Wood frogs are typically the first species that enter into the system. They, as soon as the conditions are right, the ice gets off the ponds, there are a few days of rain, all the wood frogs come, they're explosive breeders, and then their eggs are there, they leave. But what's happening is there's increased variability in the spring. The wood frogs will come, it will freeze, and all of their eggs are either frozen or they're negatively affected. So what we did was we tested how freezing or cold, then warm, then cold, then warm, influences these amphibians' susceptibility to salt. And what we found is that the variation, the freeze, the thaw, the freeze, the thaw, had negative effects, but not quite as bad as constant cold. And so what the main take-home from this study is that the variable temperatures in the early spring are having a negative effect on amphibians' ability to deal with salt. I do know, transitioning back to the concentration of salt as ponds dry, in that survey I talked to you about earlier, about the 36 ponds in Madison, what we did find, it wasn't necessarily salt, but the ponds that had the highest conductivity, so the highest salt, they had the highest degree of pathogens. So it may not be salt, but it's also having an effect on the disease dynamics. Additionally, on a more positive side, many amphibians are plastic, meaning they sense their environment and can speed up their development. So as the pond dries up, as hydro period decreases, many amphibians have the ability to allocate their resources towards development, as instead of growing bigger, they develop faster. And so that's one positive way that amphibians can deal with some of the stressors that they encounter. So if you think about environments where toads or wood frogs, they're in ephemeral ponds that dry up often. Those are typically more plastic than amphibians in permanent ponds like bullfrogs or green frogs.
I have an online question here. Won't a biodiverse host enable a virus to develop a different, more persistent type, more resistant for other bodies to fight off?
Yeah, it is possible. So the question is in a community that is more biodiverse, what about the parasite? And this is something that goes, it's a coevolution. So we, in our lab, have focused primarily thus far on the host. But to understand the effects on host dynamics, you can sort of think about a triangle. You have the host, you have the parasite, and you have the environment. Today, I talked to you about how the environment affects the host. The next step is how the environment affects the parasite, and then how the environment affects their interaction. We know that hosts can evolve tolerance to all sorts of pollutants and also their pathogens. But the pathogens can also evolve in response to the pollutant and the host. So it's a really fun thing that we get to think about, and it's something I'm excited about because that means that I'll in my lifetime, it's unlikely I'll run out of cool questions to ask.
What are you seeing as far as insecticides and how insecticides are going and affecting the amphibians? An example would be you see a lot of the yards out here up north here getting sprayed for mosquitoes. Mm-hmm. How does that affect those amphibians?
Oh, this is what I did my dissertation on. Whew. It is something that we have studied for the past 16 years. Specifically, we have studied the effects of an insecticide called carbaryl. What we have found, and on the more positive end, amphibians and actually many species can evolve tolerance to these pollutants. Evolving tolerance may allow them to persist in their environment, but what we're finding is that there's a cost to being tolerant to the pesticide. If these amphibians are allocating resources to tolerance to pesticides, they tend to be more susceptible to their pathogens. What's interesting about pesticides, there are two ways that you can become tolerant. One, you have a population, a pesticide comes in, the ones that have some trait that allows them to persist, they evolve tolerance over time. The problem with that is it can take many generations, and what happens if a population doesn't have that tolerant trait? What we discovered is that there's another way, a faster way, through phenotypic plasticity. What that means is that we found that in some amphibians, the plastic trait I talked about, they can develop faster if they encounter environment. If they encounter a predator, they change the way they look that make them more resistant to the predator. So we thought, well, why not pesticides? If they detect pesticides, can they, within their lifetime, become more tolerant? So what we did was we exposed eggs, amphibian eggs, to a very low amount of pesticides. And what we discovered is that when they became tadpoles, they were more tolerant than their siblings that were raised in pesticide-free environments. And so this is something that happens quickly within the lifetime of the amphibian. So what happens at high enough concentrations, these insecticides are lethal. It can be directly lethal or indirectly lethal, for example, by killing off through the trophic cascade that I described earlier, the domino effect. But the good news is that there seems to be multiple mechanisms that allow them to persist, with the caveat that there might be a cost to that tolerance to pesticides. So altogether, it's complex, but we're working to better understand how these different pieces all come together.
Hi. So you've been talking a lot about kind of the different life stages, and I just wondered if parasites and pollutants both maybe are more common in the aquatic settings that the amphibians are in, or the terrestrial, or if they're like facing them in both of their environments during their lifespan.
Yeah, that is something that we have been grappling with in the lab. So today, I've only talked about amphibian parasites in the water. The parasite, the fungal pathogen BD, this is a pathogen that is causing declines worldwide. And it's a pathogen that affects the keratinized tissue of amphibians, which means the skin in juveniles and adults. In tadpoles, the only keratinized tissue is in the mouth part. And so actually, BD is probably more prevalent in adult and juvenile stages than in the aquatic stages that I talked about today. The research that I talked about today focused on trematodes. These are parasites. Trematode, they have a complex life cycle. They use an amphibian, a snail, a bird. They're free swimming in the water, and they'll swim towards the amphibian. And I also talked a little bit about frog virus 3, which is an emerging infectious diseases that this is what we train our community scientists to look for. You go into a pond, and there are hundreds of tadpoles that are exhibiting, they look like they're hemorrhaging, they're red, they're upside down. So it's really particularly nasty in that system. So I think the questions, to answer your question, I think it depends on the pathogen and specifically which stage is more susceptible. But certainly there are parasites and pesticides in both systems. We focus on the tadpole system mostly because it's accessible and it's easier to contain than the amphibian, the adult and juvenile system.
So if research findings show that either a parasite or a pesticide or something else is having a very harmful effect on organisms, is there a system or process in place where you as scientists can communicate effectively to government, agencies, corporations, and the public about your findings and start the change happening?
Oh, that is a question I will ask my entire career, what the best way to do that is. I think at this point, we're not very good at it. We're amazing at doing the research, putting it into scientific articles, presenting it at scientific conferences. But when it comes to policy, and I'm talking about my realm of science, I am an ecologist. I know that there are more applied scientists out there that do the research that goes directly into the EPA. The challenge is that in order to make regulations, you have to conduct very standardized studies so that it's easy to replicate, so that you can compare across different pollutants, across different species. With our work, we're trying to integrate in this realistic complexity. And so while it's realistic, it's not something that agencies can really use because it's not standardized. And so what I try to do, and something that gives me hope, as of now, I have 3 students or colleagues that have now entered into the EPA. And so there's direct conversations about how we can be more helpful, how we can improve communication. One thing that I've learned, they open up these communication for open comments about certain pollutants. And what we can do is we can have a voice. The challenge is, as an academic, I am teaching, I am doing research, I'm doing service. And so to do that extra, it's not often something that is at the top of the priority. Whereas if a company that generates the chemical, they have people working full time, they have people paid to be able to generate those comments. So the short answer is we're thinking about it, we're trying to figure out how to best communicate up those lines, but we don't know how to do that very well yet.
Yeah, back to your comments about plasticity and juveniles being more affected.
Mm-hmm.
Okay. Are you seeing any, can you hear me? Okay. I'll repeat, going back to the comments about plasticity and juveniles being affected more than adults. Mm-hmm. Have you seen any evidence that that plastic trait is passed on to the adults? I mean, as tadpoles grow up and become adults.
Yeah. So two answers to that question. That is one of the hot questions in my field right now. So plasticity is within a lifetime, how far does it go? And then also selection or evolution on plastic individuals. So selection for individuals that are more plastic. So I'll explain that. First, if you expose an egg to low concentrations of pesticides, they become tolerant at the tadpole stage. But over time, they lose that tolerance. But if you expose the tadpole to the pesticide, then at even older stages, they become tolerant, but they lose that tolerance. And that's sort of adaptive, because if that threat is no longer there, why invest the resources to maintaining tolerance? So that's the first part of the answer. The second one is, what about across multiple generations? And this is where it's super cool in the theoretical mind that I think in, the space that I think in. You can have selection for traits that are always there. So let's imagine that this tadpole lives in an environment that is very variable. It makes sense to select for its offspring that can also be plastic, because you have no idea what environment that the offspring are going to be in. But let's say that this tadpole is in an environment where it's always consistent. Absolutely makes no sense to invest in machinery to be plastic. And so this idea of selection on plasticity versus selection on fixed traits, that's something that is really exciting and something that in my field is what we're really interested in. So yeah, thanks for the question.
I've got another online question here. Have you any examples of a parasite vanishing short of mass extinction of its preferred host?
Do I have any examples of a parasite vanishing? That's a stumper. I'm thinking about, I'm having flashbacks to my PhD days. Well, I have to defend in front of my committee, and that feels like a question that they might have asked. You know, I don't know the answer to that. I can't think of a pathogen that has disappeared. I know that populations of hosts have disappeared, but across the landscape, we have, in the pond system, these ponds are interconnected. Amphibians are moving from one location to the next. So if I had to make a hypothesis, I don't think that there would be a situation where I would expect a pathogen to completely disappear. The reason for this comes from one of my good friends' research from the University of Pittsburgh, Richard Zawacki. What they found is that in a pond, a vernal pool, this is a pool that the water will dry up by the end of the season. You have species like wood frogs that can metamorphose very quickly and get out. And so they wanted to know, would the pathogen exist in this type of pond if it dries up every year? And what they found is that green frogs, and for those of you that are familiar with green frogs, they have to overwinter to metamorphose. These green frogs are going into these vernal pools and bringing their pathogens in with them. And so that's why I think, at least in the amphibian system, in interconnected systems, I wouldn't bet against the pathogens, and I would not expect them to be completely eliminated. But just a hypothesis.
Can you hear me?
I can.
For anyone who's been here over the last decades and has property on the lake or has just been on the lakes, they've seen the water levels go up and down and go back up to a certain degree. And if you measured, on the one hand, this is a long-term cyclical factor that's been going on apparently for forever and a day. The people out at Trout Lake have an excellent graph that points this out. On the other hand, with the other factors that are coming into play, and let's just stick with road salt, over time that is accumulating. So you have a secular change and you're dealing with the cyclical change. And when you're doing experiments, how do you iron those things out and so you think you're getting accurate data and not the effect of one on the other?
I think that is going to be the challenge of the coming generation of scientists. Where we are right now is we have a fantastic understanding of individual effects, at least in my particular field. We know what this pollutant does. We know what shifting temperatures do to the system. But thinking about the experiments that we have to do to look at the interaction, it gets big very, very quickly, the factorial studies. And so lucky for us, one of the things that people are starting to do with all of these individual level studies, using tools like machine learning, can we make predictions based off of what we know on individual effects on the interaction of multiple effects? I don't think we're quite there yet, but at least that's one step that people are trying to take to tackle that problem. So I know for sure last year, the Department of Defense, they came out with a multi-billion-dollar grant for that specific purpose, for scientists to come to a solution about how to deal with multiple stressors across multiple scales. So from a small spatial temporal scale to long-term and broader temporal scales.
Well, I have one more online question here. A friend working in crayfish has found that a crayfish with a parasite behaves differently from uninfected crayfish. Do you see behavioral differences between animals living amidst contaminants compared to more pristine environments?
Yes, the answer, absolutely. So we have been, I mentioned before, we work on about 15 populations of wood frogs, and we've learned a lot about these different populations. In the populations that are living close to agriculture, they are tolerant to pesticides. Populations living far from agriculture are susceptible to pesticides. But the ones living close to agriculture are more susceptible to the parasite. We recently discovered one reason why they might be more susceptible, and it's because of this really strange behavior called necrophagy. So ranavirus, frog virus 3, it's spread through direct contact, and necrophagy is the consumption of dead conspecifics. So the populations that are tolerant to pesticides, what we think is happening is that they're resource limited, and so they're more likely to exhibit this necrophagy behavior where they then increase transmission compared to the populations that are not tolerant to the pesticides. So that's one example of how the various populations that experience various levels of pesticides have differ in their behavior that can then affect their disease susceptibility.
I have to ask, what is currently your next step that you'll be taking in your personal research, as well as what you're hoping to tackle in the future?
Oh, I have many, many dreams on the direction that I'd like to go. Where we are right now in terms of this research, we hope to take advantage of some of the genetic tools that are available to us. I talked a little bit about pesticide tolerance across these different populations. They vary in their tolerance to pesticides, to parasites. We sort of want to understand what mechanisms, what genetic mechanisms are allowing them to do this. So far, I've sort of talked about it from the individual level all the way up to the ecosystem level as we go out into the field. One of the things we're hoping to do is to dive into the molecular level now to sort of understand mechanism. What about their genetics, their physiology is allowing them to do this, and how can we use this information to understand a little bit better how other organisms deal with stress. So we're diving in deep there. On the other end, maybe some of you are familiar with the frog and toad survey in Wisconsin. One of the things that we're hoping to do on a more broader scale I have a postdoc in my lab who is incredible with generating models, predictions, and what we're hoping to do with that data set is to understand how changing climate, changing land use is affecting amphibian populations across the state. So that's sort of the interest in this area of the research. On the human side of the research, we're very interested in broadening participation. Right now, as I mentioned before, I come from a refugee family. I can't even get my parents to come out with me yet. And so the question is, how can we expand what we do to also benefit other members of society? So that's sort of in the most immediate range of things that we hope to do.
I've got one last question here that kind of follows up to that. So based on the results of your studies, will you continue art-science collaborations? And then what's your favorite piece?
Oh, I will absolutely continue art-science collaborations. And one of the things about this entire art-science collaboration that I haven't had a chance to talk about, it all started because I had an undergraduate in my lab. His mom convinced him that it was absolutely necessary to be in the sciences so that he would get a good job. And he hated everything about the lab. And so we finally had to sit down and it was like, why are you doing this? And what do you really want to do? And it turns out he is an incredible artist, and he wanted to be an artist, and he was only doing this because it was practical. And so what we ended up doing was we talked about ways we could use his passion to contribute to the sciences, and that's where this entire art-science collaboration began. He wrote a children's book. He did the art show. All of this. I take on artists now to do research in the lab because of this undergraduate. And the amazing thing is he is now a graphic designer. He runs his own company. He's part of a company, but he also does contract work, art science contract work. So if anybody is interested in this type of thing, his name is Ben McLaughlin. You can find his information on my website. He is amazing. So I do hope to do that in the future. My favorite piece. Oh man, you should see my office. I've accumulated so much amazing art. Actually, I think right now my favorite collective piece is the graphic novel. This was done by 5 undergraduates in my lab. You have storytellers, you have artists, and you have synthesizers. And I think this graphic novel, there are 6 chapters, over 100 pages. It's collectively, I think, our most comprehensive piece. So that's currently my favorite, but it depends on what I'm looking at the moment. But yeah, thanks for the question.
Please welcome me in joining, welcome me in joining. Join me in thanking Dr. Jessica Hua. Thank you.
Thank you all.
Thank you to everybody who participated tonight and to our sponsors, the Lakeland chapter of the UW Alumni Association, Rocky Reef Brewing, Monaco Public Library, UW Kemp Natural Resources Station, UW Trout Lake Station, and WXPR Public Radio. We hope to see you back here again in November for Dr. Mary Stevenson talking about sleep and health. And if you'd like to sign up for emails, you can do that with us here or on our website by clicking Contact Us. Have a good night.